Hi, I'm Srinidhi Teli, a second-year master's student from Carnegie Mellon's Biomedical Engineering Department. I've been working on dynamic engineered heart tissues, also called dyn-EHTs, to evaluate cardiomyocytes incorporated into pulsatile conduits for congenital heart disease research. Single ventricle disease is a serious congenital heart defect and is responsible for about 23% of cardiac deaths in the first week of life. Tragically, even with medical advances, less than half of affected children survive past 15 years. In the US alone, around 930 children are diagnosed with this condition each year. Our approach is to engineer heart tissues, combining cardiomyocytes and fibroblasts, the major cell types found in the heart. We then test these tissues for their ability to contract, to conduct calcium signals, which are both crucial for healthy heart function, especially under dynamic real-world conditions. Ultimately, our goal is to optimize these engineered tissues so they can be integrated into pulsatile conduits, essentially conduits that can rhythmically contract, helping us study how single ventricle disease develops, particularly in cases like hypoplastic left heart syndrome or HLHS. First, we start by differentiating stem cells into cardiomyocytes and expanding adult cardiac fibroblasts. These two cell types are essential for building functional heart tissue. Next, we seed these cells within a collagen matrix around a flexible support strip, creating an engineered heart tissue or EHT. We then culture these tissues dynamically for about 2 weeks, mimicking the contracting and relaxing environment of a real heart. This setup allows us to study the tissue during both diastole, that's relaxation, and systole, that's contraction phases. After the tissues mature, we test two main properties. First, we measure the heart strength, how much force the tissue can generate as it contracts and relaxes. Second, we measure the speed of the heart's electrical signals, using calcium imaging to determine how efficiently the signals spread through the tissue. By combining these steps, we can closely evaluate how our engineered tissues behave and how they might function in future clinical settings. The heart strength test was done by measuring its contractility. Firstly, we measured how strong the tissues were during both the relaxation phase, that's the diastolic stress, and the contraction phase, the systolic stress. The group with more fibroblasts showed higher stress in both phases compared to the collagen group, and this was true at all time points. This makes sense because higher fibroblast concentration generates more stress in engineered heart tissues because fibroblasts compact and remodel the extracellular matrix, increasing tissue density and mechanical strength. But when we looked at contractile force, which tells us how much the tissue can pull or generate force, the collagen group outperformed the high fibroblast group, and this difference was consistent over the course of the study. So next we performed calcium conduction studies. We used calcium imaging to visualize how electrical signals travel through the tissue, since coordinated signaling is vital for heart function. Our calcium wave transients and maps show that signals moved similarly whether the tissue had more fibroblasts or more collagen. The first image here represents a calcium wave transient for a field-stimulated tissue at 1 Hz. We picked 3 regions of interest from the tissue to check how the calcium wave transmitted through it, and the graphs here represent synchronous wave transmission. The second image is a calcium map for a tissue point-stimulated at 1 Hz. When we measured conduction speed, we found no notable difference between groups, meaning the engineered tissues conducted electrical signals with similar efficiency, regardless of their cellular makeup. Following this, we performed immunohistological studies. Here you can see our engineered heart tissues stained to highlight different proteins and cell structures, including actin, alpha-actinin, and cell nuclei. These stains allow us to visualize how the cells are organized and aligned within the tissue, which is critical for healthy heart function. The images show that engineered heart tissues develop well-organized sarcomeres representative of adult heart tissue. Looking ahead, we plan to expand this model by developing dyn-EHTs from Notch1 knockout HLHS iPSCs, a relevant disease mutation. We'll use these models to further evaluate contractile function, tissue remodeling, and electrical properties. By comparing these findings to control and previous disease models, we hope to deepen our understanding of how HLHS develops and progresses. In summary, our research demonstrates how EHTs can reveal key differences in contractile and electrical function depending on their cellular makeup. This platform is a step towards understanding and ultimately treating complex congenital heart diseases like HLHS. Thank you.